This paper, Finalization and Validation of Questionnaire and Algorithm of SPUR, a new adherence profiling tool, builds on two preceding papers investigating the SPUR framework. SPUR was first published in 2020 as a conceptual framework to help understand the adherence behavior of patients with chronic disease. The current paper presents the analysis of a French data set of 245 patients with type 2 diabetes. The analysis of the US dataset appeared in the article The SPUR Adherence Profiling Tool, Preliminary Results of Algorithm Development and Current Medical Research and Opinion in December 2021, and a third UK dataset is currently under analysis. The data were collected through an online survey. The survey included 4 questionnaires, SPUR, and 3 validated adherence questionnaires, BMQ, MARS, and ACCEPT. Medication possession ratio and HbA1c were also collected. Building on the preliminary algorithm created using the analysis of the US dataset, a partial credit model analysis was conducted to understand the structure of the SPUR tool and to determine whether all items reflect the same underlying concept, and then the fit of each item was examined. Principal component analysis was carried out to determine whether the data confirms to the hypothesized theoretical structure. The final structure of SPUR was determined using the hypothetical structure, the Kaiser criterion provided by the PCA, item fit, and the person-item map generated by PCM analysis. A multi-trait analysis was also used. Construct validity, concurrent validity, and known groups validity were assessed on the final SPUR questionnaire. In the end, we identified a short, intermediate, and long form of SPUR with 6, 17, and 24 questions respectively, that could be used independently or sequentially in a digital tool. We observed statistically significant correlations of medium to high size with all selected concurrent scales, while correlation between the final algorithm scores of the 3 SPUR forms and the latent trait were high. Results from the psychometric study were supportive of the validity of the SPUR tool in assessing the risk of non-adherence and the drivers of that risk. On the basis of this study, and its sister studies, we believe that SPUR is a reliable and valid tool to evaluate the non-adherence risk of patients and to investigate the behavioral drivers behind that risk.